Right now, there's a 90% chance you have at least one of these plants in your yard or neighborhood. They look beautiful. They're sold at every garden center, and landscapers install them without warning labels. But here's what the nursery industry doesn't tell you. These 7 common plants contain compounds so toxic that a single leaf can kill a small child, and their root systems are actively poisoning the soil beneath them. The most dangerous part? Most people have no idea which plants pose the greatest threat until it's too late. While gardeners focus on visible plant health, a hidden agricultural crisis unfolds beneath the surface, one that compromises soil integrity and threatens the foundation of productive growing systems. And these contamination networks operate through pathways that most homeowners never detect until severe damage occurs. Root systems function as the primary delivery system, continuously releasing compounds directly into the surrounding soil matrix throughout the plant's entire growing season. Decomposing plant matter creates concentrated toxin reservoirs as cellular breakdown releases stored compounds. Seasonal leaf drop establishes toxic layers that persist through winter months, creating sustained contamination zones that expand outward from the original plant location. Professional landscapers routinely install these toxic ornamental species without providing soil impact warnings. Garden centers stock and promote these plants based solely on aesthetic appeal and growth characteristics, while completely overlooking their chemical effects against soil ecosystems. The installation process itself becomes a contamination event, as disturbed soil mixes with root fragments and plant debris, distributing toxins throughout the prepared planting area before the first growing season begins. Cross-contamination represents the most insidious aspect of this crisis, transforming isolated toxic plants into garden-wide contamination sources. Garden tools transfer plant residues and soil particles between different growing areas. The research confirms that compounds like urushiol from poison ivy can persist on metal surfaces and clothing, spreading contamination across previously clean garden beds. Irrigation systems become distribution networks, carrying dissolved compounds through water lines and affecting soil chemistry in areas far removed from the original toxic plant locations. These plant-derived compounds create persistent contamination scenarios that resist natural soil processes. Certain toxins bind to soil particles and organic matter, forming complexes that resist microbial degradation and seasonal weathering. These compounds accumulate in soil over multiple growing seasons, reaching concentrations that affect plant growth and soil chemistry long after the original toxic plants are removed from the garden. Soil microbiome disruption represents a critical but largely unrecognized consequence of plant toxin contamination. Beneficial bacterial communities that facilitate nutrient cycling, root development, and disease resistance become suppressed in areas with elevated toxin levels. These disrupted soil ecosystems create dead zones where healthy plants struggle to establish robust root systems, leading to poor growth performance and increased susceptibility to pest and disease pressure. Current testing protocols focus predominantly on heavy metal contamination and basic soil chemistry parameters like pH levels and nutrient content. Detecting organic plant toxins requires specialized analytical procedures that can identify specific compounds at concentrations relevant to soil health and human safety. These testing methods exist within agricultural monitoring systems, but residential gardening communities remain largely unaware of their necessity or availability. The contamination cascade effect multiplies the impact of individual toxic plants exponentially. A single toxic shrub creates expanding contamination zones through root activity, leaf drop, and pruning debris that affect soil areas extending well beyond the plant's visible footprint. Cross-contamination through garden maintenance activities can distribute these compounds to vegetable gardens, children's play areas, and pet activity zones where exposure risks become significantly elevated. Detection of this contamination requires understanding the specific chemical signatures of different plant toxin families. Different compounds leave distinct molecular fingerprints in soil that persist for extended periods after plant removal. These compounds bind to clay particles and organic matter in patterns that experienced soil analysts can identify through targeted testing protocols. The complexity of these contamination scenarios demands testing approaches that extend far beyond standard residential soil analysis procedures. Understanding contamination pathways becomes essential for developing effective prevention strategies. The chemical behavior of plant toxins in soil varies dramatically based on molecular structure, soil composition, and environmental conditions. Different toxin families respond differently to soil pH changes, requiring customized treatment approaches for different contamination scenarios. Cross-contamination patterns follow predictable routes through garden maintenance activities and water movement, allowing targeted intervention strategies that prevent widespread soil contamination. This systematic approach to plant selection and soil monitoring transforms residential gardening from an aesthetic pursuit into an environmental management discipline. Professional-grade monitoring protocols adapted for residential use provide homeowners with the analytical tools necessary to detect and address contamination before it reaches critical levels that require extensive remediation efforts. Among all toxic plants, one species stands out as the most dangerous example of how chemical warfare can transform healthy garden soil into hazardous environments. Giant hogweed operates as nature's most efficient furanocoumarin production facility, manufacturing photosensitizing compounds that create severe chemical burns on contact, while simultaneously poisoning surrounding soil ecosystems through sustained toxic discharge. This invasive ornamental species, recognizable by its towering carrot-like stalks and distinctive white umbrella-shaped flower clusters, functions as a biological weapons system designed to eliminate competing vegetation through chemical warfare tactics that persist long after the plant itself disappears from the landscape. The root system operates as a continuous exudation network, releasing furanocoumarins directly into the soil matrix throughout the entire growing season. These compounds serve a specific ecological purpose, creating toxic soil conditions that suppress competing plant species while establishing the hogweed's dominance over the growing area. The chemical discharge occurs at concentrations sufficient to alter soil chemistry, transforming healthy growing environments into contaminated zones that resist natural plant establishment and ecosystem recovery. Direct root exudation represents only one pathway in the contamination cascade that spreads furanocoumarins throughout garden ecosystem. Decomposing plant matter releases concentrated toxin loads as cellular breakdown liberates stored compounds. Surface contamination occurs when sap drips onto soil during normal growth cycles, creating localized hotspots where toxin concentrations reach dangerous levels. Each decomposing leaf and stem fragment becomes a sustained-release toxin source that continues contaminating soil for extended periods. Laboratory analysis demonstrates that contaminated soil retains active furanocoumarin levels long after complete plant removal, creating persistent exposure risks that extend far beyond the visible presence of the hogweed itself. This extraordinary persistence stems from the compound's molecular structure, which binds to organic matter in soil and resists breakdown by natural soil bacteria. The binding creates stable toxin soil complexes that shield furanocoumarins from microbial degradation and seasonal weathering processes. The resistance to natural degradation processes distinguishes furanocoumarin contamination from other plant-derived soil toxins. Standard soil bacteria populations lack the capabilities necessary to break down these complex organic compounds. The molecular stability of furanocoumarins in soil environments requires specialized remediation approaches that address both the chemical properties of the toxins and the soil conditions that maintain their stability. Traditional soil treatment methods prove ineffective against these persistent organic pollutants. Garden tools exposed to giant hogweed sap transform into mobile contamination vectors that spread furanocoumarins to previously clean soil areas through routine gardening activities. Shovels, pruners, and gloves retain active toxin residues on their surfaces for extended periods, transferring contamination during normal garden maintenance tasks. The cross-contamination mechanism operates invisibly, as gardeners unknowingly distribute hazardous compounds across multiple growing areas without recognizing the contamination transfer occurring through their equipment. The contamination transfer process extends the impact of individual hogweed plants exponentially. A single plant's toxic influence spreads throughout entire garden systems, as contaminated tools carry furanocoumarin residues to vegetable beds, ornamental gardens, and children's play areas. The mobile nature of tool-based contamination creates scattered contamination hotspots that appear unrelated to the original hogweed location, complicating identification and remediation efforts. Irrigation systems become secondary distribution networks for dissolved furanocoumarins, carrying toxins through water lines and affecting soil chemistry in areas far removed from the original plant locations. The water-soluble nature of certain furanocoumarin compounds enables contamination spread through drainage patterns and watering systems. Contaminated runoff from hogweed areas affects downstream soil zones, creating expanding contamination patterns that follow natural water movement and artificial irrigation network. The compounds actively suppress native plant communities, leading to biodiversity decline in affected soil areas. These toxins interfere with seed germination, root development, and nutrient uptake in competing plant species. The suppression effect creates expanding dead zones where healthy vegetation struggles to establish viable root systems, leaving contaminated areas visibly barren or supporting only toxin-tolerant weed species. Immediate soil testing becomes essential following any suspected hogweed exposure, as visual contamination indicators may not appear until significant ecosystem damage occurs. Standard plant removal techniques prove insufficient for furanocoumarin contamination, requiring containment and disposal methods that prevent further contamination spread. The furanocoumarin contamination model demonstrates how individual toxic plants transform healthy garden environments into hazardous landscapes, requiring systematic environmental remediation approaches. Professional environmental remediation requires systematic protocols that prevent contamination spread while protecting removal teams from dangerous exposure levels. The ADDI framework, analysis, design, development, implementation, evaluation, provides the structured methodology necessary to transform hazardous plant removal from amateur gardening into controlled environmental management that protects both human health and soil ecosystem integrity. Analysis phase establishes the foundation for safe removal through comprehensive site assessment protocols that identify all toxic species present within the contaminated area. Site mapping documents the precise locations of different toxic plant species, their growth stages, and the extent of visible contamination zones surrounding each plant. Soil testing determines baseline contamination levels using analytical procedures designed to detect specific plant toxins at concentrations relevant to human safety and soil health. Cross-contamination risk evaluation examines potential pathways for toxin spread through garden tools, irrigation systems, foot traffic patterns, and natural drainage routes. The mapping process reveals contamination patterns that extend far beyond the visible plant locations. Root zone analysis identifies areas where toxin concentrations may persist in soil long after plant removal. Drainage assessment determines how contaminated runoff might affect downstream soil areas during removal operations. Tool contamination evaluation identifies equipment that may already carry toxin residues from previous garden maintenance activities. Design phase establishes the containment and safety protocols necessary to prevent contamination spread during removal operations. Containment barrier planning focuses on isolating work areas while providing effective separation from clean soil zones. Personal protective equipment specifications address the unique hazards posed by different toxic plant families, following established occupational safety principles for handling hazardous organic materials. Disposal method planning addresses the regulatory requirements for hazardous plant waste while preventing secondary contamination through improper handling. Decontamination procedure development creates systematic protocols for cleaning all tools, equipment, and clothing exposed to toxic plant materials. The decontamination specifications must address the persistence characteristics of specific plant toxins, as some compounds resist standard cleaning methods and require specialized treatment approaches. Development phase transforms the design specifications into detailed operational procedures with specific safety checkpoints and quality control measures. Removal procedure documentation provides step-by-step instructions for different toxic plant species, accounting for their unique hazard profiles and contamination characteristics. Safety checkpoint establishment creates verification points throughout the removal process where contamination levels and worker safety can be assessed before proceeding to subsequent operations. Emergency response planning addresses potential exposure incidents during removal operations. The emergency protocols must account for the specific toxins present and their physiological effects, as different plant toxin families require different medical responses. Specialized disposal container organization ensures proper segregation of different toxic plant materials and prevents cross-contamination during waste handling and transportation. Implementation requires systematic execution of removal procedures using containment barriers that isolate contaminated areas from clean soil zones. The containment system prevents toxin-laden soil particles and plant debris from spreading beyond the designated work area during removal operations. Immediate tool decontamination occurs between different work areas to prevent cross-contamination through equipment transfer. Proper protective equipment usage follows strict protocols that address the specific hazards of each toxic plant species being removed. The occupational safety mindset demands different protection approaches for different toxin families, with systematic adherence to established safety procedures throughout the removal process. Controlled waste disposal ensures that removed plant materials reach appropriate facilities without creating secondary contamination during transportation. The removal sequence follows predetermined patterns that minimize worker exposure while preventing toxin spread through soil disturbance. Plant cutting techniques avoid creating aerosols or dispersing toxic compounds onto surrounding soil areas. Root system extraction employs containment methods that prevent contaminated soil from mixing with clean areas. Evaluation involves comprehensive post-removal assessment to verify successful contamination, elimination, and ecosystem recovery. Post-removal soil testing confirms that toxin levels have decreased to acceptable limits following plant removal and any necessary soil treatment. Contamination monitoring tracks toxin persistence over extended periods, as some plant-derived compounds remain active in soil long after plant removal. Ecosystem recovery assessment evaluates the return of beneficial soil microorganisms and the successful establishment of replacement vegetation in previously contaminated areas. Protocol effectiveness analysis examines the success of containment measures, decontamination procedures, and safety protocols to refine future removal operations. The ADDI protocol transforms dangerous plant removal from haphazard garden maintenance into professional environmental remediation that ensures complete contamination control. This systematic approach provides the framework necessary to address the most persistent contamination scenarios, where individual plant species create lasting environmental hazards that extend far beyond their visible presence in residential landscapes. Among the most deceptive threats, oleander's extensive cultivation throughout residential and commercial landscapes has established millions of contaminated soil sites, where cardiac glycosides operate as persistent environmental toxins affecting human health, pet safety, and fundamental soil ecosystem processes. This evergreen shrub produces concentrated levels of cardiac glycosides in all plant tissues, compounds so potent that ingestion of a single leaf contains sufficient toxins to cause fatal cardiac arrhythmia in small children and household pets. The plant's extensive fibrous root system functions as a continuous toxin distribution network, releasing cardiac glycosides through root exudates throughout the entire growing season, while simultaneously absorbing and concentrating these compounds from decomposing plant matter in the surrounding soil matrix. Seasonal leaf drop creates expanding contamination zones as fallen foliage decomposes and releases concentrated glycoside loads directly into surface soil layers. The decomposition process actually increases toxin bioavailability, as cellular breakdown liberates stored compounds that bind immediately to surrounding organic matter. Cardiac glycosides demonstrate remarkable binding affinity for soil organic matter, forming stable chemical complexes that resist natural degradation processes occurring in healthy soil ecosystems. These toxin soil complexes persist for extended periods following plant removal, creating persistent environmental reservoirs that continue posing exposure risks long after visible. Oleander presence disappears from the landscape. The molecular structure of cardiac glycosides enables strong chemical bonding with organic soil components, effectively protecting these compounds from microbial degradation. Soil contamination testing conducted around established oleander plantings reveals active glycoside concentrations extending well beyond the plant's visible drip line, demonstrating substantial root zone contamination that affects far larger soil areas than most homeowners recognize. The contamination pattern follows root distribution networks, creating underground toxin reservoirs that remain invisible until specialized soil analysis reveals their presence and concentration levels. Irrigation systems can transform into contamination distribution networks when they service oleander-planted areas, potentially carrying dissolved cardiac glycosides through water lines to previously uncontaminated soil zones. The water-soluble nature of certain glycoside compounds enables their movement through watering systems and natural drainage patterns. This contaminated water flow can affect edible plant cultivation areas, creating potential exposure pathways when toxin-contaminated water reaches vegetable gardens and fruit tree root zones. Composting operations involving oleander debris create highly concentrated toxin sources that multiply contamination risks exponentially compared to fresh plant material exposure scenarios. The composting process accelerates cellular breakdown while concentrating cardiac glycoside in the resulting organic matter, producing finished compost with toxin levels significantly exceeding those found in living oleander tissues. Gardeners unknowingly distribute these concentrated toxin sources throughout their properties when using contaminated compost as soil amendments or mulching materials. Soil microbiome analysis reveals that cardiac glycosides severely disrupt beneficial bacterial communities essential for healthy nutrient cycling, root development, and natural plant disease resistance mechanisms. These disrupted microbial ecosystems create degraded soil conditions where replacement vegetation struggles to establish viable root systems and maintain normal growth patterns. The suppression of beneficial soil bacteria reduces organic matter decomposition rates, nitrogen fixation capacity, and phosphorus availability, fundamental processes required for healthy plant establishment and sustained growth. The bacterial community disruption extends beyond immediate toxin exposure areas through contamination spread via garden tools, foot traffic, and water movement patterns. Contaminated soil particles carried on equipment surfaces introduce cardiac glycosides to previously healthy soil areas, gradually expanding the zones where beneficial microorganisms face suppression from toxic compound exposure. Oleander contamination scenarios require immediate plant removal combined with soil replacement in high-traffic areas where human and pet exposure risks reach dangerous levels. Complete soil excavation becomes necessary in children's play areas, pet activity zones, and edible plant growing areas where cardiac glycoside contamination poses direct health threats. Long-term monitoring protocols track glycoside persistence in remaining soil areas, as these compounds maintain biological activity for extended periods following initial contamination events. The cardiac glycoside contamination model demonstrates how popular ornamental plant selections create lasting soil health problems that extend far beyond aesthetic landscaping considerations. The widespread cultivation of oleander throughout residential neighborhoods has established extensive contamination networks affecting soil quality across millions of properties, where homeowners remain completely unaware of the persistent environmental toxins present beneath their landscaping choices. These contamination networks expand through pathways that most gardeners never recognize during routine maintenance activities. A single contaminated pruning shear transforms into a mobile toxin distribution system capable of spreading plant-derived poisons to dozens of previously clean soil areas during routine garden maintenance sessions. The contamination transfer operates invisibly, as gardeners unknowingly carry urushiol from poison ivy, alkaloids from nightshade plants, and cardiac glycosides from oleander across their entire property through normal tool usage patterns. Standard gardening practices amplify contamination exponentially. Each cut, dig, and cultivation activity becomes a toxin distribution event that establishes new contamination hotspots throughout previously safe garden areas. Urushiol demonstrates extraordinary persistence on metal tool surfaces, maintaining active contamination potential for extended periods following initial poison ivy contact. The molecular structure of urushiol enables strong chemical bonding with steel, aluminum, and iron surfaces commonly found in garden tools, creating persistent toxin reservoirs that resist standard cleaning protocols. Contaminated tools transfer active urushiol to soil during routine cultivation activities, establishing new poison ivy contamination sites in areas that never hosted the original plants. The contamination transfer occurs through direct soil contact as tools disturb earth, and through plant contact as contaminated implements touch healthy vegetation. Research confirms that urushiol can persist on clothing, shoes, tools, soil, and animals that have made contact with the plants, creating multiple pathways for contamination spread throughout garden systems. These persistent residues bind chemically to both metal tool surfaces and fabric fibers used in gardening gloves and clothing. The adhesion properties enable toxins to persist through multiple garden sessions despite standard washing and cleaning attempts. The persistence creates cumulative contamination as toxin concentrations build up on frequently used tools over extended periods. Standard cleaning methods prove insufficient against these persistent organic toxins because they lack the chemical specificity necessary to break the molecular bonds between toxins and tool materials. Soap and water remove visible plant debris but leave invisible toxin residues that maintain biological activity. Standard cleaners and detergents may actually enhance toxin binding to certain metal surfaces by altering surface chemistry in ways that increase molecular adhesion. The cleaning failure creates false security as gardeners believe their tools are decontaminated when active toxin residues remain present. Contaminated irrigation equipment operates as a distribution network that spreads dissolved toxins through water systems, affecting soil chemistry across entire garden beds and potentially reaching edible crop-growing areas. Sprinkler heads, drip emitters, and hose connections retain toxin residues that dissolve into irrigation water and deliver contamination to previously clean soil zones. The water-based distribution system enables toxin spread across much larger areas than direct tool contamination. As irrigation networks service entire landscape systems, dissolved toxins travel through pressurized water lines and emerge at distant irrigation points. Clothing contamination creates mobile toxin sources that operate independently of tool-based transfer mechanisms. Plant residues embed in fabric fibers during normal gardening activities, creating wearable contamination sources that deposit toxins through contact with soil, plants, and garden surfaces. The fabric binding enables toxin transport between different garden areas as contaminated clothing moves throughout the landscape. Synthetic fabric materials demonstrate particularly strong binding affinity for certain plant toxins, creating persistent contamination that survives standard washing cycles. Vehicle tires and garden cart wheels function as contamination transport systems that move toxin-laden soil particles between different garden areas, expanding contamination zones far beyond original toxic plant location. The tire treads collect contaminated soil during routine garden access and deposit these particles in parking areas, pathways, and previously clean garden zones. Garden cart wheels operate similarly, picking up contaminated debris and soil particles and distributing them along travel routes throughout the property. The mechanical transport system creates scattered contamination patterns that appear unrelated to original toxin sources. The contamination cascade operates through garden systems with each contaminated tool, piece of clothing, and equipment item serving as a distribution vector for toxic compounds. The multiplication occurs because each contaminated item creates multiple new contamination sites during normal use, and those sites become sources for further contamination spread. A single pair of contaminated gloves can establish toxin presence across dozens of garden areas through routine planting, weeding, and maintenance activities. Understanding these contamination transfer pathways becomes essential for maintaining soil health and preventing the establishment of garden-wide toxin distribution networks that compromise entire landscape ecosystems. The contamination patterns extend beyond immediate tool contact, creating complex seasonal cycles where toxin levels fluctuate based on plant growth phases and environmental conditions. Popular spring bulbs operate as underground toxin manufacturing centers, producing alkaloids alkaloids that cycle through soil systems with seasonal intensity patterns that create persistent contamination challenges throughout residential landscapes. These beloved spring flowers function as chemical laboratories, continuously manufacturing compounds capable of causing seizures, organ failure, and cardiac arrest, while simultaneously releasing these toxins into surrounding soil through multiple contamination pathways that operate throughout the entire growing season. Licorine from daffodil bulbs leaches continuously into soil through root systems that deliver alkaloid compounds directly into the surrounding soil matrix during active growth periods. Research confirms that daffodils and related plants contain licorine, which can cause vomiting, drooling, convulsions, and irregular heartbeats, with the highest concentrations of toxins found in the bulbs themselves. Decomposing bulb scales create additional contamination sources as outer protective layers shed and release concentrated toxin loads into surface soil layers. The decomposition process liberates stored alkaloids at concentrations exceeding those found in living bulb tissues, creating persistent alkaloid reservoirs that maintain biological activity long after the visible flowering period ends. The alkaloid release process operates through root cells that actively transport licorine from bulb storage tissues into surrounding soil. This release serves multiple biological functions, deterring soil-dwelling pests, suppressing competing vegetation, and establishing chemical territory around individual bulbs. The continuous discharge creates expanding contamination zones that grow larger each growing season as bulb colonies mature and establish deeper root systems. Colchicine contamination from autumn crocus operates through different mechanisms, with the alkaloid persisting in soil and creating stable contamination that resists natural degradation processes. Research confirms that autumn crocus contains colchicine and other alkaloids, which are toxic to people and pets, causing oral irritation, vomiting, shock, and organ damage. The molecular properties of colchicine enable binding with soil components, creating persistent toxin reservoirs that maintain biological activity throughout winter dormancy periods and into subsequent growing seasons. Soil conditions influence colchicine persistence, with the compound remaining active through seasonal changes and moisture fluctuations. These persistence patterns create contamination levels that fluctuate throughout the year, with toxin availability changing based on environmental conditions that affect chemical mobility within soil systems. Bulb division and naturalization multiply contamination zones exponentially as mother bulbs produce offset bulbs that establish in previously clean soil areas, expanding the overall toxin footprint annually through natural reproductive processes. Each new bulb becomes an additional toxin production facility, manufacturing and releasing alkaloids into soil areas that previously supported healthy plant growth. The naturalization process creates expanding contamination networks as bulb colonies spread through underground division and seed production. The annual expansion follows predictable patterns based on species characteristics and growing conditions. Daffodil colonies expand outward from original planting sites through bulb division, creating ring-shaped contamination patterns that grow larger each year. Autumn crocus spreads through both bulb division and seed dispersal, creating scattered contamination hotspots throughout garden areas where seeds germinate and establish new bulb colonies. Soil conditions influence alkaloid mobility and persistence, with different environmental factors affecting toxin availability and potential for contamination spread. Moisture fluctuations drive toxin movement through soil layers, carrying dissolved alkaloids deeper into soil profiles during wet periods and concentrating them in surface layers during dry conditions. Pet exposure incidents spike dramatically during bulb flowering seasons, when alkaloid concentrations in soil reach annual peak levels and contaminated soil particles become airborne through wind dispersion and gardening activities. The flowering period coincides with maximum alkaloid production, as bulbs mobilize stored energy reserves for reproduction, creating the highest toxin concentrations in both plant tissues and surrounding soil. Contaminated soil particles attach to pet fur and paws during normal outdoor activities, creating direct exposure pathways that pose serious health risks to household animals. Understanding seasonal toxin cycles becomes essential for implementing effective soil monitoring protocols during peak alkaloid production periods, when contamination risks reach maximum levels. The cyclical nature of bulb contamination requires timing soil testing and remediation efforts to coincide with seasonal alkaloid fluctuations. These individual toxin sources create complex soil chemistry scenarios, but the contamination challenge intensifies dramatically when multiple toxic plant species establish within the same garden areas, creating contamination interactions that exceed the individual effects of single plant toxins. Foxglove and nightshade represent two of the most dangerous plant families commonly found in residential landscapes, each producing distinct classes of toxins that create severe soil contamination challenges when established within garden environments. Foxglove manufactures cardiac glycosides, including digoxin and digitoxin, throughout all plant tissues, while nightshade species produce various alkaloid compounds that affect nervous system function and cellular processes. Both plant families release their respective toxins into surrounding soil through root systems and decomposing plant matter, creating persistent contamination zones that pose serious health risks to humans and pets. Cardiac glycosides from foxglove demonstrate remarkable persistence in soil environments, binding strongly to organic matter and resisting natural degradation processes that normally break down plant-derived compounds. These glycosides affect heart rhythm and cardiac function at extremely low concentrations, making soil contamination particularly dangerous for children and pets who may come into contact with contaminated earth during normal outdoor activities. The compounds leach continuously from living root systems throughout the growing season, while decomposing plant matter releases concentrated toxin loads that accumulate in surface soil layers. Nightshade alkaloids operate through different contamination pathways, with compounds like solanine and related alkaloids creating toxic soil conditions that suppress beneficial soil organisms and interfere with normal plant growth processes. These alkaloids affect nervous system function and can cause severe gastrointestinal distress, respiratory problems, and neurological symptoms in humans and animals exposed to contaminated soil. The alkaloid contamination persists through seasonal changes, maintaining biological activity in soil long after visible plant material disappears from garden areas. Soil ecosystems demonstrate severe disruption when either plant family establishes within garden environments. Beneficial bacterial populations, essential for nutrient cycling, organic matter decomposition, and healthy plant root development become suppressed in areas where cardiac glycosides or alkaloids reach significant concentrations. The bacterial suppression creates degraded soil conditions where replacement vegetation struggles to establish viable root systems and maintain normal growth patterns. Earthworm populations provide visible indicators of soil ecosystem health in contaminated areas, with significant population reductions occurring in zones affected by either foxglove or nightshade contamination. The decline in earthworm activity signals broader disruptions in soil food webs and nutrient processing systems that extend far beyond the immediate toxic effects of individual plant compounds. When both plant families establish within the same growing areas, the contamination effects intensify as different toxin classes accumulate in shared soil matrices. The combined presence of cardiac glycosides and alkaloids creates more severe soil ecosystem disruption than either compound class produces individually. Mixed contamination sites exhibit dramatic alterations in fundamental soil chemistry parameters, with pH levels and nutrient availability becoming severely compromised. Contaminated runoff from areas containing either foxglove or nightshade carries dissolved toxins to downstream soil zones, creating expanding contamination patterns that spread far beyond original plant locations. The water-soluble nature of certain glycosides and alkaloids enables their transport through natural drainage systems and artificial irrigation networks. Surface water contamination occurs as dissolved toxins migrate through soil layers and potentially affect water sources used for irrigation throughout landscape systems. Soil testing protocols require specialized analytical methods capable of detecting both cardiac glycosides and alkaloid compounds at concentrations relevant to human and animal safety. Standard soil analysis procedures designed for heavy metals and basic chemistry parameters cannot identify these organic plant toxins, necessitating specialized testing approaches that target specific compound classes. The testing must account for seasonal fluctuations in toxin concentrations as plant growth cycles affect contamination levels throughout the year. Professional remediation of foxglove and nightshade contamination demands comprehensive approaches that address the unique chemical properties and persistence characteristics of both cardiac glycosides and alkaloid compounds. The contamination scenarios require understanding how different plant toxins behave in soil environments and selecting appropriate treatment methods based on the specific molecular structures and binding properties of each toxin class present in contaminated areas. Effective decontamination requires understanding how different plant toxins behave in soil environment and selecting treatment approaches that address their unique molecular characteristics and persistence pattern. Alkaloids bind differently to soil particles than cardiac glycoside, while compounds like furanocoumarins resist degradation through entirely different mechanisms. Standard soil remediation methods designed for heavy metal contamination or petroleum products prove inadequate for plant-derived organic toxins because these biological compounds operate through sophisticated molecular interactions that conventional treatment approaches cannot address effectively. Professional soil decontamination services become essential for severe contamination scenarios where toxin concentrations exceed safe exposure limits or where multiple plant toxin families create complex contamination profiles. These services employ specialized knowledge, advanced techniques, and appropriate equipment to manage hazardous materials effectively. Professional assessment determines which treatment approaches will prove most effective based on specific toxin types, soil conditions, and contamination severity levels. Activated charcoal applications represent one approach for addressing certain plant toxin contamination, though effectiveness varies significantly based on toxin molecular structure and soil conditions. The treatment process requires precise application rates and careful consideration of soil chemistry factors that influence binding effectiveness. Soil conditions dramatically influence treatment outcomes, with different environmental factors affecting how well various approaches perform in specific contamination scenarios. Biological treatment methods utilize specialized approaches that target specific plant toxin classes through natural degradation processes. These methods require careful monitoring of environmental conditions and may extend over several months as biological systems establish and begin systematic toxin processing. The treatment timeline depends on toxin types, soil conditions, and environmental factors that affect biological activity levels. Soil excavation and replacement delivers the most reliable decontamination results for severe contamination scenarios, where other treatment approaches prove insufficient. Complete soil removal eliminates contamination sources while providing immediate protection from ongoing toxin exposure. The excavation process requires containment protocols that prevent contaminated soil from spreading to clean areas during removal operations. Proper disposal of contaminated soil follows hazardous waste protocols that classify plant toxin contaminated material based on specific toxin concentrations and exposure risks. The disposal process requires specialized transportation and delivery to approved facilities equipped to handle organic toxin contamination. Standard disposal methods remain inadequate for severely contaminated soil that poses ongoing environmental and public health risks. Phytoremediation utilizes toxin-tolerant plant species that can absorb and process specific plant toxins through their root systems over multiple growing seasons. The process operates gradually as remediation plants establish root systems and begin systematic toxin uptake from contaminated soil. Different plant species demonstrate varying effectiveness against specific toxin classes, requiring careful selection based on the contamination profile present in affected areas. Chemical neutralization methods target specific toxin classes through molecular reactions that break down toxic compounds or convert them into less harmful derivatives. These approaches must avoid creating secondary contamination through harsh chemical applications that damage soil ecosystem health. Each treatment requires careful selection based on toxin molecular structure and existing soil chemistry conditions. The inadequacy of standard cleaning methods becomes apparent when dealing with persistent plant toxins that resist conventional soil treatment approaches. Simple washing or standard soil amendments cannot address the molecular binding properties and chemical stability that enable these toxins to persist in soil environments. Professional assessment becomes necessary to determine appropriate treatment strategies that can effectively neutralize specific contamination scenarios. Successful decontamination protocols often combine multiple treatment approaches tailored to specific toxin profiles, soil conditions, and contamination severity levels. The selection process requires understanding how different treatment methods interact with various toxin classes and soil chemistry parameters. Professional soil decontamination services provide the expertise necessary to evaluate contamination scenarios and implement systematic remediation processes that transform contaminated sites into safe, productive growing environments. The decontamination process establishes the foundation for long-term soil recovery, but verifying successful toxin elimination requires systematic monitoring approaches that can detect residual contamination and track ecosystem restoration progress over extended periods following remediation efforts. Systematic monitoring protocols establish the framework necessary for verifying successful decontamination and tracking ecosystem recovery following toxic plant removal operations. The monitoring process transforms soil remediation from uncertain guesswork into verified environmental restoration, providing quantitative evidence that contaminated areas have returned to safe, productive growing conditions. Professional testing services provide the specialized analytical capabilities necessary to detect residual plant toxins and measure biological recovery indicators that signal successful ecosystem restoration. Baseline soil testing establishes the fundamental reference points necessary for measuring decontamination progress throughout extended monitoring periods that can span multiple years following toxic plant removal operations. The initial testing protocols must identify all present toxin classes, measure their individual concentrations, and document contamination distribution patterns across the affected soil areas. These baseline measurements provide the quantitative foundation for tracking toxin degradation rates and verifying the effectiveness of remediation efforts over time. The testing procedures require specialized analytical methods capable of detecting plant-derived alkaloids, glycosides, and organic toxins at concentrations relevant to human safety and soil ecosystem health. Regular soil sampling operates through systematic collection procedures that track toxin degradation rates across multiple soil depths and geographical zones within the contaminated area. And the sampling protocols identify areas where contamination persists despite remediation efforts, revealing hotspots that require additional treatment or extended monitoring periods. Each sampling event measures changes in toxin concentrations, soil chemistry parameters, and biological activity indicators that signal ecosystem recovery progress. The sampling timeline captures seasonal variations in toxin mobility and degradation rates that fluctuate with temperature, moisture, and soil chemistry changes. Soil biological health serves as a critical indicator system that reveals ecosystem recovery status through the gradual return of beneficial bacterial populations as toxin levels decrease below critical suppression thresholds. The biological analysis tracks the diversity and abundance of nitrogen-fixing bacteria, phosphorus-mobilizing microorganisms, and organic matter decomposers that form the foundation of healthy soil ecosystems. These bacterial communities demonstrate predictable recovery patterns as soil toxin concentrations decline, providing early indicators of successful decontamination before chemical testing confirms complete toxin elimination. The biological monitoring process reveals whether soil conditions support sustainable plant growth and natural nutrient cycling processes essential for long-term soil health. Beneficial bacterial diversity increases as toxin levels decrease, creating measurable indicators of ecosystem restoration progress. Professional soil testing services can assess bacterial community health through specialized analysis that determines whether soil biological systems have recovered sufficiently to support healthy plant establishment. Plant growth testing utilizes sensitive indicator species selected for their ability to detect residual contamination levels that may persist in soil after apparent decontamination completion. These living detection systems respond to trace toxin concentrations through stress responses, growth inhibition, or physiological changes when exposed to residual plant toxins. The testing protocol involves growing sensitive plants in soil samples collected from different areas within the remediation zone, with plant growth rates and development patterns providing measures of soil safety levels. Groundwater monitoring prevents contamination spread to adjacent properties through systematic testing of water sources and soil moisture at various depths below the surface contamination zone. The monitoring network ensures that plant toxins do not migrate through soil layers and contaminate groundwater systems that serve neighboring properties or natural water sources. Water sampling protocols detect dissolved alkaloids, glycosides, and other mobile toxin compounds that may leach downward through soil profiles during precipitation events. The groundwater testing program establishes monitoring points at strategic locations around the contamination perimeter to track toxin movement patterns and verify containment effectiveness. Seasonal monitoring captures changes in groundwater toxin levels that correlate with precipitation patterns and soil chemistry fluctuations. The water analysis protocols employ the same specialized testing methods used for soil contamination assessment. Post-removal monitoring extends for predetermined periods based on the specific toxin classes involved and their known persistence characteristics in soil environments. Different plant toxin families require varying monitoring durations, with some contamination scenarios necessitating extended surveillance programs to ensure complete toxin elimination. The monitoring timeline adapts based on ongoing test results that track actual toxin degradation rates in specific soil conditions. Professional verification protocols confirm complete contamination elimination through multiple testing methods that provide independent confirmation of soil safety levels. Chemical testing, biological analysis, and plant growth assessments must all indicate successful decontamination before monitoring programs conclude and soil areas receive clearance for unrestricted use. This comprehensive verification process provides confidence in long-term soil safety and productivity, establishing the foundation for implementing systematic garden management strategies that prevent future contamination scenarios. Toxic plant contamination represents a critical threat to modern agricultural productivity and soil ecosystem integrity that demands immediate systematic intervention. These 7 dangerous plants operate as contamination factories that transform healthy garden soil into hazardous environments through continuous toxin release, root exudation, and seasonal debris accumulation. The contamination extends far beyond visible plant locations, spreading through irrigation systems, garden tools, and natural drainage patterns to affect entire landscape ecosystems. Begin with comprehensive garden assessment to identify all toxic species present and map contamination zones requiring immediate attention. Contact professional soil testing services to establish baseline contamination levels before implementing removal protocols. Systematic removal requires specialized containment, decontamination, and disposal procedures that prevent toxin spread during extraction operations while protecting human health throughout the remediation process. The agricultural implications extend beyond individual properties, as contaminated soil compromises food production capacity and threatens sustainable growing systems essential for modern agriculture.